Okay, so I'm going to give you an example on my work. This is part of my dissertation when I was in Canada. So there was a big study in Ontario a long time ago, like before Jeff and I went to Canada. So that was in 1990. So they had this study. They were looking at 400 patients, and they got samples from atypical males. Yung mga ganyang itsura yung sa picture. And then a lot of isolates were taken, and they were most of the isolates were identified as dermatophytes like trichophyton or trichophyton, microsporum. But there were, because hundreds of isolates were done, but out of the hundreds of isolates, there were 5 that they couldn't identify. So next slide, please. So ito yung 5 isolates. Nilagay nila, dineposit nila sa UAMH, doon sa University of Alberta Microfungus Collection. And sabi ng supervisor ko, Connie, you work on these ones. I'm gonna, but it's not part of my thesis. Sabi ko, no. And so she said, but no, you work on this one as a side study. So I did. But what happened was, it was very unusual, because when you infect the nail, you're supposed to be keratin degraders, because the nail has keratin, and they were eating the nails. But when I tested them, they were not keratin degraders at all. So I get, okay, ano to mga to? Are these dermatophytes or non-dermatophytes? Kasi they were looking like they are really true pathogens. Because there were other cases, not only from this 5. So then I started my study, and then what I did was, so here's what it looked like. These are the anamorph states of those 5 fungi, okay, or the 5 isolates. So you can see there are different types of anamorphs. So yun pa lang, nalilito na ako, okay, are they the same species? So ito yon. So I had to do my questions when I did my proposal. So you have to present your proposal sa iyong panel. So, okay, these are my questions. Are these 5 isolates the same species? Are these 5 isolates dermatophytes? Are these 5 isolates true pathogens? Yung mga ganon. Then I have to go and answer those by doing the experiments. So what I did was, at that time, so I did mating studies. So in all combinations, so I kinross ko sila lahat. And then I did also DNA sequencing. So sabi ng supervisor ko, this is part of your thesis. Kung hindi pwede, kasi magme-mate ako eh. Hindi ko alam kung kailan mangyayari yan. Kasi minsan mga fungus, mga ulit yan. Pagka sinabi mong mag-mate kayo, ayaw mag-mate. Tapos pag ayaw mo namang pag-matein, tsaka naman produce ng ascospores. Nakakainis talaga. So anyway, that's what I did. It almost cost me my PhD kasi 8 months na, naghintay na ako. This is my last paper, my last article to publish, and then I can have my book. Hindi pa rin, 8 months na. So I said, no, I'm gonna give up. Kailangan itapon ko na kayo. Ang dami na hundreds of plates. But, sabi ko, sige, itatapon ko na kayo today. Pero before that, I will look at you. I will look at these plates one more time. Pagtingin ko sa mga plates, after 8 months, Yahoo moment na naman. Kasi nakita ko na yung mga ascospores at saka ascomata. Natakot sila kasi autoclave ko na sila. So then,
ayaw nila titang makremate.
I know! So then, ang nangyari, when I saw those ascospores, it reminded me of arachnomyces. Kasi these ascospores are what they look like with the arachnomyces theliumorph. So then, Na-connect na namin ngayon na meron kaming bagong anamorph sa Arachnomyces theliumorph. So what I did was, okay, but then when we did the sequencing, it was distinct from the other species. So what do? We have to describe this as a new species. So then I named it Arachnomyces canei after the great mycologist in Canada, Dr. Julius Kane. He wrote a book on dermatophytes. And then the anamorph name was Onychocola canei. Ang galing ko, di ba? Kasi pareho sila ng epithet. At least, hindi ako masamang mycologist. Hindi ko nililito yung mga tao. Kasi pareho sila. I mean, you can actually think right away, oh, ito yung anamorph ng Arachnomyces canei. So, okay. So now we have a new species. We have to publish it. Pero anong lineage nito? Yung lineage, for example, genus, anong sunod? Yung family, anong family to under? Anong order to under? Anong, kung Ascomycota ba to, ganyan. So alam namin Ascomycota siya. But we don't know whether it is, which order it is in. So nung ginawa namin, nagawa na naman kami ng phylogenetic assessment. Ito yung next one. So when I did my phylogenetic assessment with this, together with most of species, like representative species in the phylum Ascomycota. So malaking tree ito. If you see that it is, so we assumed that it was in the Onygenalis, but it's not. It's outside. It's its own distinct group. And then it is also sister to the Urothialis. So what do we have to do? Sabi ko, well, because if we don't erect an order and family, our species will be incertisadis, meaning unknown, undefined. Wala siyang place. Wala siyang lugar. So, hindi pwede yon. We have to erect an order and a family. So that's how it came to be. So that we accommodate our species. So we had to erect an order and a family para merong bahay yung aming species. Okay? So the next one is now we publish it. So we have this one, Arachnomycetalis, So Arachnomycetalis is the order. So you see, A-list is the suffix of orders. ACE or CE is the suffix of family. So Arachnomycetalis, Arachnomycetaceae, and then the authority, which is us. Okay. And then we also published the species in another article with the span of, with my supervisor and my panel. Okay. So next one, please. So since I became a mycologist in 1998 until last month, I have discovered 16 species of fungi, and I have erected or established one order and one family. Okay, next one. So for you guys, if you have names of fungi, if you want to check whether the name of the fungi is still current, you can go to these websites and check from there. And also it's very good to do, to look at publications, because usually the publications will be very, very up to date. Sometimes Index Fungorum is not up to date because the people who are working there are only voluntary. So kung kailan lang sila may time. Okay. And then also, if you want to look at the next one, JP, look at names of Penicillium and Aspergillus, you can go to this website because the names there will be correct. Because there are many names now that are not Aspergillus anymore. So if you want to be sure, go to this website and they're always updated. So here is Index fungorum. So as you can see, the name listed in blue, for example, Neosartoria fisheri, is the old name, and the one in green is the current name. So that's how you check for the current names. Okay. So now, is it the lab's role to report name changes to the clinicians? Absolutely. It is our role to do that because we are the ones who are studying them. We are the ones who are looking up these things. So it is our responsibility. Thank you.